All right, so let's dive right into it. Aiming for like 15, 20 minutes or so for the talk, and then some time for questions and comments at the end. So today we're talking about orthology and paralogy at the Alliance. As Carol mentioned, my name's Chris. I'm a biologist and a developer at the Alliance of Genome Resources. So here's what I'm going to talk about today. We're going to start with the source data, where the orthology and paralogy come from at the Alliance. Then we're going to move on to the implementation for the orthology and the paralogy, talk a little bit about how both of those data are integrated with other Alliance data sets that we have. And then finally, I have a slide on data accessibility, so how to download this data, these data for your own use. Okay, so before we even show the website itself and the orthology paralogy tables, let's talk a little bit about the source data. So we don't use a single algorithm at the Alliance for determining orthologous and paralogous relationships. We rely on DIOPT, and DIOPT is a tool developed by the DRSC, Integrative Orthology Prediction Tool here, and there's a citation on the screen as well. It's an excellent paper. I recommend giving it a read. And DIOP works by using a combination of algorithms from various sources, and then the recommended orthologs or the recommended paralogs are a count of those algorithms. So if a certain number of algorithms call a certain result, then that becomes the higher result, and then the less a result is called by a group of algorithms, the lower it falls down down of our recommendation on the table. So we'll see a picture of that, what it looks like in a minute or so. So DIOP tries to strike a balance between casting a wide net and specificity. So different algorithms work in different ways, and the thought being that if we look at many algorithms at once and kind of do a meta-analysis on the results, then we get a better result. Okay. So here's a list. These are all the algorithms that we're currently using at the Alliance. So the algorithms that we pick from for orthology are different than paralogy, but there's a very significant overlap. There's a few small changes. Some of these algorithms have data that's supplemented by curators themselves. So we have the data from groups that specialize in running that algorithm, but then that's also complemented with data that, let's say, a ZFIN curator might provide, and that would be manually curated orthology. So if we look more in detail at our orthology list here, the ones that I've highlighted in red, those sources of orthology are complemented by biocuration. So biocurators have looked at some of these relationships between genes and provided directly evidence that we incorporate into our algorithm. So this would be different than just using the output from the groups that run these algorithms normally. An important point here is that the underlying data, so there's a unified UDAPROT reference proteome that's used for this, and that originates from the Quest for Orthologs group. So there's an agreed upon set of data that all these algorithms are running, so that at the very end we get a more consistent result. And that wasn't something that was present when this was all first started a long time ago, but it's something that's come about as the project has matured. So it's there's a link there for Quest for Orthologs if you want to read about it, and also the reference proteome at EBI. And the other point on this slide is, as I mentioned before, apart from the calculated computational results, we have the curated sets provided by individual groups as well. If we look at our algorithms for paralogy, it's a smaller set. We're not using the Quest for Orthologs data for paralogy. We're using the set that's from DIOP, that's derived from their pipeline without the Quest for Orthologs. And in this case, as you can see, there's biocurated data presented from 3 groups, and then for the paralogs, we just have SGD providing additional paralogy data that we use in our algorithms. So that being said, let's look at what the table actually looks like, and let's talk about the implementation that we have here now that everyone is aware of the way we use these algorithms. So if you load up a page here, this is the indie genes from Drosophila. Right on the left side of your screen, you'll see the first 2 results under the summary, or are the orthology and paralogy sections on our gene report. So this is for the gene report. If you click on orthology, you'll jump down to the orthology table. So here's what it looks like. It's iterated a few times since we first released this many years ago. We're going to talk about some of the things on this page and on how they function. And first, I'd like to talk about what's at the top here. These are the filters that we provide. Oh, actually, before I jump into that, you can see on the right side of the screen, let me mention this. This is a better segue from the algorithms talk I just had. So we'll talk about the algorithms first, actually. So you can see this list on the right side of the screen, and it's all the orthology or algorithms that I had on the previous slide. An empty box means that that result was not called by that algorithm. So like for the first example there, you can see that Ensemble didn't call the Homo sapien ortholog there, so the box is blank. If there's no box, that means that that information was not available from that algorithm. So for example, ZenBase at the very right of this column here, ZenBase is not providing human orthologs for a fly gene, which would make sense because they're the frog database, so they wouldn't be doing that. Neither is ZFin. So those two columns at the very end are blank because we don't have the data it's missing, which is different than the data was there potentially and not called as a match from the algorithms. Okay, so there's your list of methods on the right side. And then what I wanted to focus on next is talking about these filters. So the filters at the very top of the screen here, you have three options to choose from. They default to a stringent filter. So we have stringent, moderate, and no filter. And there's a pop-up there that explains these, but I have a slide for it on the next slide. This gives the people that are visiting the website, the users, a way to reduce the size of this list by leaving it on stringent, or expand this list and look at the possible results by going all the way to no filter, which will just show us all the orthologs that are possible, even ones that have very low counts. Okay, so the pop-up has a lot of words. It's on the right side of the screen right now. So that's, if you want to mouse over that question mark and read about the filters on the website, feel free. All the information is right there. The left side is my attempt to provide cliff notes for how these filters work. So the stringent filter is our strictest filter. So the primary rule, if you're going to show up as a result on the stringent filter, is you have to be called by three methods. And you have to be either a best match forward, which means you're the best match for the algorithm going from the target gene to this from the source gene to the target gene, or a reverse match. So a reverse match means that you can see it says best reversed here on the screen. It's the column next to best. So that means that if you were to flip this around, so if you started at, for example, SLC 13A3, then you're a best match coming back to the gene that you're starting from. So we have best as in we're going from our source gene to our target gene for orthology, and we also have best reverse, which means the opposite is also true. So if you were on that other page, it would be the best option to come backwards to. So that indicates a very strong orthologous relationship. So you'll show up in the stringent filter if you're called by three methods and you're either the best forward or reverse match. And then we really trust our curators here. So if you were included in the predictions from any of our hand curated methods here from ZFin, HGNC, or ZenBase, and the HGNC hand curations are actually from RGD. So they say HGNC, but they're really from RGD for the human rat to human orthology calls. We're going to update that in the near future to actually say RGD, but we haven't gotten around to that yet. But that's just a little caveat that I wanted to mention. And it's also included if the best match is true in both directions. So those are the three criteria. So if your best match and reverse match together, that's good. If you're called by the other two things that I just mentioned as well, you show up in the stringent filter. Moderate, we relax this a little bit. So just being called by three methods, regardless of whether you have best match in either way. Then we keep the special cases, excuse me. So those recommendations will always show up. And then no filter is just everything. So you can get quite a lot of results on no filter. And we like this because, you know, orthology is difficult. As anyone who works in the field will tell you. I mean, there's been some wonderful progress, especially over the last 10 years to tackle this issue. But if you want to look at everything, then we provide that. You can just click no filter and see all the results and make the choice for yourself. Again, so just bring you back to the screen for another preview before I move on of how the orthology page works. I should also mention quickly, we have a link to Panther gene trees when available for genes. So if you were to click that link, you can see it has a little leaving the site arrow on it. You can go to Panther and look at the orthologist, the gene tree view of this gene. So I'm not going to really talk about that much, but I recommend trying it out to learn about all the data from Panther because it's very interesting and useful. Okay, and I already talked about the best and best reverse, so we'll just go to the next thing. Right. So I wanted to show an example of the ZFin being used here. So you can see on the right side of the screen now the ZFin boxes are present and checked. which means that the ZFIN curators provided some information. So that's a contrast to this previous one where you see ZFIN is not populated. There's a interesting result that you can get when you go to some of our Zenbase gene reports here, like this one. So this is not a bug. This is a Xenopus laevis page, and you can see that we don't have much information here. In fact, the only information we have for orthology for this particular gene comes from Zenbase. So this is a kind of a limitation in the diopt approach that we have right now. We only have a certain number of species that are run through this very long pipeline to generate the results, starting from the quest of orthologs all the way through the assembly of the data. And laevis in this case doesn't fit or doesn't exist in the starting criteria of organisms or species that we're working with. So these data here are provided by the Zenbase curators, which is fantastic because it gives us this extra bit of information for orthology, and it does allow us to connect this species back to other species. But it is limited. The only thing we have here, as I mentioned, is the data from the Zenbase curators. And if you look at Tropicalis, you can see that this is actually included in the full set of diop species that are run. So we have a much more complete data set, but we don't have from diop Tropicalis to laevis. So that is missing. And in this case, you can see, again, that's provided by the Zenbase curators. So if you're browsing the site and you're looking at some of our frog genes here, and you see this kind of interesting result, that's the reason why it exists. And it is nice that we have the ability of the system to incorporate the curator provided data here so that we can expand it past the diop set of species that we normally work with. Okay, great. So that's the orthology overview that I wanted to give with the table. And we're going to move on to look at paralogy real quick here. So this is an example, again, for the gene Indy of the paralogy table, and it's a little bit different than our orthology table. The principles are still largely the same, so you can see the methods on the right side again. You can see the list of algorithms that we have available. Excuse me. There's a few new columns. So we have rank, just like we kind of have rank, but we call it count on the orthology one, but there's a special calculation for rank, which I'll talk about momentarily. We also have alignment length, similarity, and identity. So we have some things that we don't have on the orthology table. And you can see we do have method count directly on the right side there, which matches this count here. Okay, so I want to talk about rank because that's a bit important. And this was something that we came through trial and error and some feedback from curators on how to make this work. If you mouse over the rank question mark there, you can read our definition of rank here. So we created this formula to kind of properly, rank, for lack of a better word, the results here for paralogy, because we felt that there was more information other than the count that would be useful for people, especially with identity and similarity and the alignment from these particular results in paralogy. It's a little more complicated than just an orthology count because of the relationship and the way paralogy, I guess, is as a research topic in a field. So this was mostly trial and error. So we came through, we went through a lot of discussions with biocurators. We looked at people's genes that they're very familiar with. We did some calculations to see whether the results are, look acceptable to us or not. And through many trial and error, we ended up with this weighting system, which informs this rank that we have here. And again, this is not, there's no perfect solution to this. There's no perfect answer for this, but this was kind of the best that we came up with after a very lengthy discussion with people who are very experienced in the field. So rank is calculated with this algorithm, and you can, again, see the results that you can see here. And you can see that, this is a good example for Indy and Indy 2 because there are two very similar results here with the same method count, but we are ranking one higher than the other based on the increased similarity here between the one. And, all of this, again, is transparent. So you don't have to take our word for the rank being the way that it is. We encourage people to look at the results and judge for themselves too, especially in cases like this one that I showed where the results are actually very close. Okay. Okay, great. So now that I've talked about the orthology tables, the standalone tables, I want to talk about the other three parts of the gene page where we incorporate orthology data and integrate it to other data sets that are available at the Alliance. And the first one I'll talk about is the Go annotations. So on any gene page here, if you go to the function Go annotation sections of the page, there's a checkbox which is unchecked by default, I believe. It says compare ortholog genes. If you check that, then this table will make a call out, it'll populate with our orthologous genes, and that's from diopt. But then it will also make a call out to the API at Go and retrieve all of the annotations for those orthologous genes. So it takes a couple seconds to make that call. But then in the end, you end up with this rather beautiful chart here of your starting gene at the very top here. So this example is with Notch from Drosophila. And then you can see all the orthologs for Notch. And then you can also see all the conserved annotations or related orthologous annotations, I guess you can call them, between all of these genes here that are being called as orthologous. And you can change the stringency. By default, it's set as stringent. You can also change the species. And then there's an extra checkbox underneath, which you can see, which is to show functions that only have at least one experimental evidence. You can remove other forms of evidence from this and just show, I want to see things that are functionally backed up here with experiments. So that's really useful. And then from this chart, you can click and go to the other genes as well. The second one is disease association. So the top looks very similar here. You have a checkbox. This one's checked by default. You have the stringency and the species selectors. And this is showing you the annotations for disease annotations now across orthologous species. And you can also choose to include negative annotations, which is unchecked by default. So it's very similar to our Go chart here, but also extends to disease annotations.
Great. So what's the color for?
The color is for the number of annotations. So the darker the color, the more annotations exist for that.
You mean child terms of the Go terms?
Yeah, yeah, I believe so. Yes.
No, Chris,
yeah.
it's data.
It's data supporting that functional annotation, right?
Yep.
Yeah.
So it's the publication count?
Not necessarily. Annotation count would be more accurate, right?
Yeah.
Okay.
Annotation count, yes.
Because you could have multiple annotations from a single publication, so it's probably more accurate to say the actual annotations.
So those are the slim terms. Go slims.
Yes,
Yeah.
as
Okay.
I believe someone with more experience.
Yes. Yeah, so those are the slim terms, and it's the sum of all of the annotations for all of the descendants of that slim term.
Okay. Yeah.
Thank you.
Chris, there was another, there are a couple questions in the chat. Do you want to wait on those, or do you want to address them as you go?
I could address them. I don't have the chat, how.
So one is about, so zebrafish has duplicated genes. How does Alliance decide one-to-one orthology between human and that's
yeah.
one.
Okay, I see them now. Claire, do you want to answer to that? Because Claire's on the call. Claire Hugh, she is the reason this project exists and works the way as well as it does. Claire is the one who handles the upstream diop processing for all these data, so she's fantastic. I mean, Claire, do you have an answer for that? I'm not really familiar with how it's handled, the duplication issues.
Yeah, we do. When you remember, you put if it's the best match, forward reverse. There's a yes, right? And in diop we also have yes adjusted, like for zebrafish, because we basically worked with Zfin to find out all the gene duplication. If those genes are not the best match, we say it yes adjusted, even though it doesn't have the best diop score.
Right. So there's basically adjustments made in consultation with the Zfin curators to pick the best.
Exactly.
Right.
But only based on their duplicated gene annotation, not based on their regular ortholog annotation file. We have a different file for that. But I think I also annotated that for you for the AGR assembly with yes adjusted. Yeah.
That's correct.
Chris, sorry, could I just say one more thing? I want to emphasize that we're not trying to enforce a strictly one-to-one orthology in all cases, that we allow for this kind of ambiguity from going from one species to the other, especially with these recent duplications. So so whereas if there's one best to best reciprocal best match, then that sounds like a good candidate for being a one-to-one match. But we don't guarantee from one species to any other species that there'll always be an exact match. Here that you can see there's 3 SLC genes that are orthologous with the same algorithm count, 2 of which are both best reciprocal blasts. You might ask, how do you get best reciprocal blasts from more than one? I think it's about homology matching thresholds, and saying that we can't, in a lot of cases, strictly make a one-to-one call.
Yeah, and I picked this one on purpose because it's messy. And you know, it kind of goes with the mentality of here's everything we have. And to answer Joel's question, I see in the chat too. Yeah, our recommendation, we really don't, like Chris was just saying, we don't want to force anything on anyone. Like some cases, like Indy, for example, there might not be a best quote unquote. So it's kind of an exercise left to the reader to take the data here and best interpret it for yourself. And we've had many lengthy conversations within the Alliance on how to handle this, and that's kind of where we've left it. Because
So
it's a
then, if I can ask, have you
Yeah.
considered making the counter argument, which would be making a somewhat definitive statement that there is no one-to-one match between these two organisms for this gene?
We could say that, I suppose, like we could write that out. I think the way we have it now, it's maybe not the most self-explanatory from the table itself because there's several options that, you know, it says best 3 times. But you know, to that point, I think Joel, I think that's a good point because we could definitely put that in the mouse over description to explicitly say that that's the case. I think that would be a useful thing to inform the users. Chris, were you going to say something too?
Yeah, I wanted to just say that I think it comes down to a matter of thresholds. If you wanted to be, as long as the protein products of some of these genes weren't actually identical, in which case there's absolutely no way algorithmically to determine that which protein is more analogous or orthologous. You could probably nitpick and you know, get down and split hairs to the finest grain of detail, and then it would be kind of an arbitrary decision. Not arbitrary, but you'd have to decide a bunch of different parameters as to which is the best way to call it. And when it's that close, I think we don't offer to say that it is the best one-to-one ortholog, but we also don't go as far as to say there is no one-to-one ortholog. But as Chris said, I think this provides the information that users can use to make their own judgment calls.
And I guess that also in the chat now, Baha has another question. And, you know, to speak to what Chris Grove just said, I guess our confidence is just that it's more like, here's the data, and then you can make the choice based on what we have. I mean, we're confident in the use of the reference proteome. There's been a lot of work put into that. We're confident in the quality of the algorithms. But at the end of the day, when it comes down to, you know, here's the list, here's what they say, it's definitely an exercise left to the reader to be satisfied with what the selection is. And we don't want to endorse or, you know, any particular result as being the one result, because I think in this part of biology, that's a very strong claim to make, and we're careful not to really make that when we can. That makes sense. Just head here. So we just have a couple more slides here. The conversations are fantastic, by the way. Thank you for the questions and the input. This is excellent. The last example here I just wanted to show. So we were just at the disease annotations associations, and this one is expression. So it's very similar to the disease, but we also have orthologous genes for our expression. And you can see the one difference here is there's a red slash through the box here. So if the other species is lacking the structure or developmental stage to make the comparison, then it's not included. But these are the same things. So it's the SLIM system with the number of annotations for that orthologous species indicates the color here in the box. So similar to the other two graphs that I showed. So if you'd like to get the data, if you'd like to get these data, so we have the orthology section on the downloads page. You can go to the downloads page. This is on the top left here. We have it available in TSV and JSON. And then there's a couple ways to get it also from our API. In the Gene API, you can get the orthologs and paralogs for any of these pages that I just showed. And then we actually have an older homology. Sorry for the confusing name, but we call this is from many years ago, and it needs to be updated, and it will. The homologs endpoint here, where you can get kind of more detailed results between species or their methods, or using the taxon ID. But I think for most people, just getting the orthologs for a gene on the top right here, using that Swagger API gene endpoint would probably be sufficient. Okay, so with that, we're reaching about 20 minutes or so. So the next webinar is August 21st, which would be presented by Chris Mungle to talk about ontologies. So feel free to join that. The registration is through the events page on the Alliance Genome Resource website. So please go and register for that if you're interested. The acknowledgements, Claire, as I mentioned, is right at the top. I cannot overstate the work that Claire has put into this project, the amount of emails that have gone back and forth to make this possible, and her dedication to it is greatly appreciated. We just simply wouldn't have it without her. And also the DRSC. There's been a number of members on our Orthology and Paralogy working group over the years, and they're listed here, and also people that aren't officially on the group, like Chris, who was participating, and Sue, apologies, I know you provided much feedback here, so your name should be next to Chris. So everyone, please imagine that Sue Bello is written next to Chris here on the acknowledgement slide. And then the PIs and our steering committee and our funding sources. So thanks. Any additional questions? Oh, I see one just pop up there. So the species, that's a great question. There's more species available via DIOP than we currently show. So we have one for every species at the Alliance right now. And we don't have, as I mentioned earlier, the Laevis, which is we only have Tropicalis for the Xenopus, not the Laevis. But I think all total, there's, Claire, is it 40 or 50 now species that you have in DIOP, or am I being too generous? How many species are in it at this?
Our current main database, we have 11 or 12,
Oh,
but we also have a sister database focused only on like insect, like and the ticks.
Right. That's the new one.
Yeah,
right.
but that one we haven't released to public yet. Yeah, we're working on that one. So, because it's not easy to scale up at the DIOP. So our strategy is to building the satellite databases with different focus.
Does orthology genes show similar expression in homologous? I'm not sure. That's a good question. For the expression annotations we have, and maybe someone who's worked on them could say, I don't know if we're taking in cell type expression for many of the species that we're using. I mean, like cell culture, do you mean? Or I guess just cell type would be, yeah, that would be a thing, obviously.
I think the comparison is done using the Uberon ontology for anatomy that is supposed to be, so every Alliance member that submits expression data submits the expression pattern, the localization, as well as a mapping to the upper level Uberon anatomy terms that actually represent the components of the ribbon, the major categories. So if it was hepatocyte or liver, it might be involved in the liver or digestive system or some sort of similar processing. However, the liver's processing gets bundled together. It would fall under that same category.
Thanks, Chris.
And there's a question about how confident the Alliance is in one-to-one orthology between mouse human, human rat, and mouse rat. And I think the answer to that is that we show all of the lines of evidence there. So it's up to the user in many ways to decide how confident to be.
Chris, I do have a question about the expression. What does that color reflect for the expression?
I believe it's the same as the other ones. It should be the number of annotations for that particular anatomical structure or stage or things of that nature. So the more annotations of that expression, the darker the shade is.
Yeah, but this is a fly example. If you look at the mouse example, it says MGI, and also say other sources, GEO, single cell expression atlas. I don't know exactly what does that mean. So yeah, here, what exactly used for the color coding? Is that the primary source?
Oh, which ones? I think that's a good question. I think it's let's see.
Chris, this is Daniela, if I may.
People that have worked on it, yes.
The primary source of data for Flybase is the one that is represented in the color-coded ribbon.
Okay.
So the darker the color, the more annotation, as it was for the other data types. And the primary source that you have right on top, it links back to the original Flybase data.
Mhm.
Then the other sources are other expression sources for third-party resources that we thought they were valuable, and they are hyperlinked. But the annotations that you see in the ribbon, they are the actual data coming from Flybase, from the annotations from Flybase.
Yeah, yeah, that's good. I think it's good to have some document to show
Yeah, I'm
exactly
not
what is the color coding is for.
So it
It's
does say at the
written-
very bottom.
Yeah, if we go to, let's see, is my screen still showing?
Scroll
Yeah,
down. Yeah.
we could see it at that last slide, but you can see it better here.
Okay.
It says cell color indicative of annotation volume at the bottom of the ribbon.
But I wonder, do we explicitly say expression in orthologs? I don't know if we explicitly say that it's just the primary source, like Claire was asking. And I think that would be because if I didn't, I mean, if I didn't know and say, okay, maybe you integrated sources from Geo as part of this ribbon, it would
Yeah.
clarify that. And we
Right.
should make that
We
change, I think.
should, yeah, we should
Well, and-
clarify the text.
Yeah, but in the table, it does tell you what the source of all of the data is, like for each row.
Huh?
So the table the table below the ribbon is the data from the ribbon from wherever you've
So
clicked.
if I click on these things, you can see how this
Oh.
table is refreshing here with more information. And since everything is coming from, these sources, then it should show the source here that you
Yeah.
can find. I mean, that being said, it might be nice just to add one sentence in here.
Yeah, I think
Yeah.
that'd be great because usually people will mistaken the expression level. Actually, this is a annotation number, right? It's
Yes.
more a qualitative. You reflect the qualitative expression. It's not quantitative.
Yeah, it's,
Like yeah, you know what I mean. So it's good
Well,
to show it.
yeah, it's intended to, I think, be a proxy for confidence, as in number of independently observed
Yeah, yeah,
experimental evidence.
exactly. It's not the absolute level.
No.
Right. It's not a level of expression, it's the number of
Yeah, yeah.
evidences for that annotation.
Exactly, yeah.
Yeah.
This also appears to be primarily at a tissue level for the most part. Are there plans in place to try to carry this down to the, I mean, it's a big project, I know, so I'm asking you to do like 10 years of work, but there are approaches that have been already started to do this kind of work.
Yeah, and Joel, I know MGI has been working on some of the cell type specific. So I think as each mod can get more into that, we'll get more of that data in.
Yeah.
I
It's
think-
hard to map cell types cross species. Probably it's even more difficult.
Yeah, definitely.
Yeah.
And there's one more quick question. I just wanted to show here that we do have all the three Go, so biological component function. We have it separated by the Go process here on this graph, so you can see how we've categorized it in three different components.
And when you click on the table below, it should give you the,
Yep.
or if you do all annotations, I think, if you click on all annotations, then it'll say aspect, and that's just function, process, component.
You can see the other ones here. Yeah.
Yeah, awesome.
I think I wanted to just make one more point about the expression is that, Daniela from Wormbase could correct me. Well, I guess I have 2 points. One is that predominantly, if I'm not mistaken, these individual rows represent individual annotations, and these are mostly low throughput expression analyses with reporters, et cetera. And so they're not necessarily quantitative for the most part. Whereas the high throughput data, expression data from GEO or Expression Omnibus, and other resources, we don't count individual annotations from those. I think there's probably a plan to incorporate kind of more quantitative aspects from high throughput studies. And getting to the single cell stuff, I know Danielle and others at Wormbase have been working out many different ways to display some nice innovative ways to display single cell expression data and to show clusters, taking into account the statistics that come along with like RNA-seq data of single cells. So we don't have that in the Alliance currently, but I think several members of the Alliance have been working independently on different ways to accommodate visualizing that data. And I think the plan would be to eventually harmonize those processes as well for that data.
And can I just clarify that this is all low throughput. None of it is RNA-seq based.
Yep.
And I can tell you that because that's all that RGD has for rat. And so that's why you don't see any rat annotations there, because everything that we have is RNA-seq based.
Yeah, so there's a kind of an inherent partition between the two classes of data that aren't fully integrated, but due to the difficult nature of that type of integration, but could be thought about in the future.
And the large amount of data that will be included when we start bringing in RNA-seq based data.
Mm-hmm.
So I just have one comment about this expression data. So when I think I don't know, a few months ago, I downloaded the expression data from AGR, and it's more like a very the data in fine resolution. It's not something organized like this. So is that possible to have something equivalent to this table, make it available for people so people can easily make the call if this gene is expressed in muscle or in fat body or not? Just right now, if I download the expression file, the FTP file, it's just it's just the resolution is just too high. I'd like to get access to the low resolution result.
I think sorry, Chris, if you don't mind. I think that
No, this is a very useful discussion. Please continue.
we have to review the degree to which all different types of data in the Alliance are integrated into Alliance Mine, which is the Alliance instance of Intermine. But I think that would probably be the best place to go for like breaking it out. Because I imagine what you're saying, when you download the file, you get probably like a list of a whole bunch of different tissues for one gene.
Exactly.
Yeah.
It's very difficult to use, but I think the table here, basically you already group all the data. You basically annotate the data to lower resolution, right? The big tissue type, then you display it here nicely. I just wonder if you can make that result
Chris,
directly available to people.
It might be a fairly easy thing to do to add a column to that download, which is just the list of header terms to which the annotations fall. So right now we have the exact term.
Exactly.
But if we
yes.
added another column which says actually it goes with this slim term as well, that might be an easy way to get what Claire wants.
I see. Yeah.
Yeah, the file generator would be easy to modify for that if that would be useful. We can definitely look into that and make a ticket for that.
Yeah, we're going to be revisiting that whole download file generator soon, so we can certainly include something like that. It makes a lot of sense.
Yeah, thank
Maybe
you.
we'll talk offline about that, Claire, from where.
Sure.
And I really appreciate, I just want to say real quick before we wrap up, all the feedback from the other Alliance curators here, because this is a very complicated project. And I'm certainly not the only person involved with this. So thanks for all the conversations and the information.
Well, and thank you, Chris, for a great presentation and lots of great discussion. As Chris mentioned, our next office hours is August 21st. It will be on ontologies in the Alliance, presented by Chris Mungle. We'll take a break in July, so we won't have a July office hours, but we'll pick up again in August and then again in September. So stay tuned for that, and thank you all very much for joining us today.
Thank you.
Thank you. Bye.
Thank you.